Module 13, the digestive system. Accessory organs, the liver, pancreas, and gallbladder. Up to this point, we've gone over how the food travels throughout the gastrointestinal tract. But there are also organs that are exterior to this route that play an important part in the digestive system. Let's first discuss the liver. It's the largest internal organ, and it has a great deal of digestive responsibilities. Perhaps its major job in digestion is to produce bile. Remember that bile is a chemical that helps to digest fats. Once it produces bile, it stores it in the gallbladder, where the bile becomes concentrated. It is then ready to be secreted into the small intestine when it's required. The liver is composed of two lobes, left and right. Because it's such a large organ, it has its own major artery that supplies the cells of the liver with oxygen and nutrients. The hepatic artery brings in blood to the liver. But there's also another large blood vessel that reaches the liver. The hepatic portal vein brings blood there. Why are there two vessels that supply the liver with blood? Well, remember, with the exception of the pulmonary arteries, all arteries bring oxygen-rich blood away from the heart and to other areas of the body. So the hepatic artery brings oxygenated blood from the heart to the liver to supply its cells with oxygen. But the hepatic portal vein brings blood from the small intestine, stomach, and spleen, and large intestine to the liver. This blood has already given up its oxygen to other areas of the body, but it does have lots of nutrients in it because they were primarily absorbed through the small intestine. So the liver takes these nutrients to process them. That's one of its jobs. Now, liver tissue can be divided into lobules. Each lobule has a defined structure. They are hexagonal shaped. Each corner of the hexagon has a portal triad made up of two blood vessels and a bile duct. One of the blood vessels is from the hepatic portal vein, and one is from the hepatic artery. The bile duct collects bile and sends it to the common hepatic duct. You may realize I'm using the word hepatic often here. Hepatic means associated with the liver, so the hepatic artery is an artery that is involved with the liver. The center of each liver lobule has a central vein which eventually joins with other veins in the liver and forms hepatic veins that continue to join together and empty into the inferior vena cava, which sends blood to the heart. Hepatocytes are functional liver cells. Remember, hepatic refers to liver, and cyte means cells. These liver cells extend outward from the central vein in thin cords, like the spokes of a bicycle wheel. Because they are cord-shaped, they are called hepatic cords, and each of these cord-shaped cells is separated by widened blood vessels called hepatic sinusoids. The hepatic sinusoids are full of phagocytic cells, and we'll talk about what those cells do in a bit. Now, the hepatic portal vein brings nutrient-filled blood to the liver, though it doesn't have much oxygen any longer. It branches into smaller and smaller veins, and each becomes a part of a portal triad. And as I mentioned earlier, the hepatic artery brings oxygen-rich blood to the liver from the heart. As it enters the liver, it also branches into smaller and smaller vessels, each eventually becoming a part of a portal triad. Then the blood of both of these types of vessels mixes up in the hepatic sinusoids, so the hepatocytes can get the oxygen and nutrients that are necessary to survive. The hepatocytes also have the job of taking many of those nutrients and processing them for other jobs throughout the body. Next, the blood collects in the central vein, where it eventually travels to the hepatic vein and leaves the liver as it moves to the inferior vena cava. Now there's one more vessel in the portal triad I need to discuss. It's the bile duct for the collection of bile. You see, hepatocytes produce bile and then secrete it into the bile canaliculi in the hepatic cords. It then moves into the bile ducts within the portal triad. These ducts join together with the hepatic duct. The bile then moves to the cystic duct and then on to the gallbladder. So bile flow moves in an opposite direction than blood flow within a lobule. Blood flow moves from the edge of the lobule where the portal triads are to the center of the lobule at the central vein. But bile moves from the inside of the lobule to its edges where the portal triads are. Bile production is a major job of the liver. It's made by the hepatocytes or cells of the liver. Then it travels to the gallbladder. Now, bile doesn't digest food. It doesn't have any digestive enzymes, but it does play a major role in digestion. Its job is to help to neutralize acid from the gastric juice. You see, bile is very alkaline. It's the third secretion in the digestive system to neutralize the acidic chyme. It also helps to emulsify fats in the chyme. To emulsify something means to break it up into tinier parts so it's more evenly distributed throughout a material. So chemicals in the bile, called bile salts, work on the large drops of lipids, separating them into smaller bits. That's helpful because the enzymes that work to digest fats are water soluble. Thus, they can only work on the surface area of a fat droplet, unable to dissolve within it. So the smaller the fat droplets are, the more surface area there is for the enzymes to break the lipids down, and thus fats are digested more quickly. If the bile duct were to become blocked, then bile couldn't emulsify the fats, and much of the fat would be undigested, being lost to the body and traveling with the feces. Additionally, lots of unwanted substances in the body are removed by the hepatocytes and are integrated into the bile. A good example of this is cholesterol. Cholesterol can be secreted into the bile as a waste product. Another major job of the liver is to process nutrients coming in from the intestines. Let's say, for example, there is too much glucose in the blood. The hepatocytes will take it in and alter it into glycogen, which is a polysaccharide. Glycogen is the polysaccharide that animals make in order to store extra glucose in the body. So the liver behaves as a storage area for extra starches. Another way the liver processes nutrients coming in from the intestines is to change some nutrients into other nutrients. This again is done by the hepatocytes, and the process is called nutrient interconversion. One type of nutrient conversion is when monosaccharides such as fructose and galactose enter the liver. They are changed into glucose. Thus, that is why blood sugar is pretty much always glucose. Another example is with phospholipids. Remember that phospholipids are the main chemicals that make up cell membranes. The hepatocytes combine fats and other digested chemicals into these useful molecules. So far, I've talked about how the liver produces bile, stores glycogen, processes nutrients from the intestines, performs nutrient interconversion, and it also plays a role as one of the main chemical manufacturing facilities in the body. It makes lots of blood coagulation factors, such as prothrombin, fibrinogen, and others. This is a synthesis function. The hepatocytes are the workhorses that perform all the liver functions I just mentioned. They compose over 99% of the liver. Another function of the liver is phagocytosis. This is due to the hepatic sinusoids with their phagocytic cells. Remember, phagocytic cells are white blood cells that help the spleen break down old erythrocytes. Phagocytic cells also get rid of old white blood cells, bacteria, and other unused materials within the blood. Lastly, we can say that the liver detoxifies the blood. You see, lots of processes in the body produce byproducts that could become toxic if they build up into higher levels. A good example of this is ammonia, created as a result of amino acid breakdown. If ammonia was allowed to build up in the body tissues, it could be toxic. So the liver chemically alters ammonia into urea, a smaller molecule that isn't as toxic to the body tissues. Urea is eventually removed by the kidneys and leaves the body via the urine. It's now time to discuss the gallbladder functions. It's pretty easy to discuss now that we've gone over all the functions of the liver. The bile the liver produced leaves the liver via the common hepatic duct. It then goes to the gallbladder through the cystic duct. Now, it doesn't move out the common bile duct because there's a sphincter muscle sealing it off from the duodenum. So the bile backs up in the cystic duct and causes the gallbladder to swell as it fills up. As long as bile is in the gallbladder, salt is moved into the gallbladder wall via active transport. Then water moves out by osmosis. This makes the bile more concentrated, making it more powerful. So when the intestine produces a hormone called CCK, the sphincter that keeps the common bile duct closed relaxes, and the gallbladder contracts. That causes bile to move through the duct into the duodenum. Now, though the gallbladder is important in the digestive process, a person can live without one. Sometimes the gallbladder or its duct becomes plugged by gallstones. Gallstones are made primarily of cholesterol and are the size of small peas. You already learned that bile can contain cholesterol, and the gallbladder removes water from the bile. If enough water is pulled out, the cholesterol sometimes can become solid and form a gallstone. Some gallstones will remain in the gallbladder and are harmless, But if one moves into a duct and plugs it up, it can be a very painful situation. Sometimes these gallstones can pass through the duct on their own. But if not, surgery may be required to remove the gallbladder so the situation doesn't occur again. If the gallbladder is removed, then bile will constantly flow out of the common hepatic duct into the common bile duct and into the duodenum. The final accessory organ of the digestive system is the pancreas. You already learned some information about the pancreas when we discussed the endocrine system. But the pancreas also plays a role in digestion. So, though the pancreas is an endocrine gland, I'm now going to talk about its exocrine functions. Exocrine glands secrete chemicals through the ducts. The pancreatic duct joins the common bile duct and empties into the duodenum. The pancreas secretes over a liter of pancreatic juice every day. This juice contains bicarbonate, so it has a basic pH. And that's a good thing because it's able to help neutralize the acidity of the chyme coming out of the stomach. In fact, pancreatic juice is the fourth alkaline secretion that neutralizes the chyme. Pancreatic juice also contains several enzymes. Trypsinogen, chymotrypsinogen, and procarboxypeptidase are enzymes that aren't active. That's because they digest proteins. And you already know that protein enzymes cannot always be active or they will begin to break down the organs that secrete them. So trypsinogen is activated into trypsin when it reaches the small intestine. This is done by enterokinase and other trypsin molecules I already talked about when I discussed the small intestine. Then trypsin activates chymotrypsinogen into chymotrypsin, and it activates procarboxypeptidase into peptidase. Then trypsin and chymotrypsin start breaking down proteins into peptides. Peptidase will finish the protein digestion by breaking down the peptides into amino acids. Another enzyme in pancreatic juice is amylase. Amylase breaks down polysaccharides into maltose. Now, maltose is a disaccharide made up of two glucose molecules, so it's broken down into the glucose molecules by maltase, made by the intestinal gland. Lipase is another pancreatic juice enzyme. It breaks down triglycerides into fatty acids. In fact, this is a very important enzyme. When you see the other parts of the alimentary canal, such as the mouth, stomach, and small intestine, they all have enzymes to break down protein and carbohydrates, but the pancreas is the only organ that makes enough lipase to digest fat. Thus, if the pancreas is not working correctly, the body will have a difficult time digesting fats. There's one final point I want to make about the types of food you digest. Think about when you eat an apple. The apple is made of cells, so you're eating those cells. As you already know, cells contain lots of nucleic acids in the form of DNA and RNA, and those molecules are gigantic. They have to be broken down during the digestive process, so enzymes called nucleases are made and secreted by the pancreas so that these nucleic acids can be properly digested. Module 13. The digestive system. Experiment 13.2. Histology of the liver. In this experiment, we're going to take a closer look at liver tissue. Now, the slides that we'll be looking at are a compilation of a number of different pieces of tissue stuck together. So we're probably not going to see the liver lobule hexagonal shape, but we will be able to see what the tissue looks like. So let's go ahead and take the human liver tissue cell and put it under the scope at 40 power magnification and focus in. And you want to be looking for a white circular area. Once you find a white circular area, center on that, and that's a central vein. So you want to put that in the center of your field of view and increase the magnification to 100 power. And let's go ahead and center and focus on that. All right, once we're looking at that, take a look at the tissue that's surrounding that white hole. It's very different from the tissue surrounding it. The tissue surrounding the hole is part of the wall of the central vein, and so it's got a different makeup. Now, take a look at the tissue that's away from the central vein. Those purple dots in the nuclei are the nuclei of the hepatocytes. So you'll see some purple dots. The nuclei took the stain a little differently, and so they got darker. And then in between the hepatocytes, you're going to see winding canals of white going back and forth. And those are the hepatic sinusoids and the bile canaliculi. at this magnification, go ahead and draw a picture of what you're looking at, including the central vein with its cells of its wall and the hepatocytes and all of the different parts that we've discussed just now, making sure you've labeled those. Now at this point, keep the magnification at 100 power, and you want to take a look around your slide and move it around slowly, and you want to look for a couple of smaller holes in the tissue. Look for a grouping of 3, and those 3 holes near one another. If you can find one of those on yours, go ahead and center that. That's a portal triad. So I'm going to try to look at that for one of those on my slide. And here we go. And go ahead and focus on that. All right, and make a illustration of that at 400 power magnification. So once you've found it, center on it and magnify that to 400 power and make an illustration of the detail of the cells in that triad. Once you've completed that, you can clean everything up and you can put it away.